You guys are all right? Sujit, do you have an okay with you?
Yeah.
All right, great. All right, good evening, everybody, and welcome back again to the new edition of the postgraduate psychiatry lecture series organized by the Department of Psychiatry, DY Patil Hospital, Navi Mumbai. And as we've always endeavored to do, we always try to bring you new topics, new perspectives, and new speakers who are stalwarts in their own right. And we also have added this feature of having a chairperson who's also an academic luminary. So they add a new perspective to the whole attempt of academic excellence that we're trying to achieve. We are extremely pleased to have with us today Dr. Biju Viswanath, who is a known figure on the lecture circuit. And obviously, Dr. Sarkil will introduce him in detail. And we have Dr. Sujit Sarkil, who is with us today, who will be chairing the session and moderating the session. And we are grateful for the both of them to have taken time out from their schedules and been with us today. It gives me pleasure to introduce Dr. Sujit Sarkil. He is an associate professor at the Institute of Psychiatry at Kolkata. He is an editor of the Bengal Journal of Psychiatry. He is an associate editor at the Indian Journal of Psychiatry. He is an assistant editor in the Indian Journal of Psychological Medicine, also at the Indian Journal of Private Psychiatry. He has more than 60 publications in national and international peer-reviewed journals, and his areas of interest are suicide, OCD, child and adolescent psychiatry, and genetics. He's also been on various academic committees of the IPS. I've had the pleasure of being on a couple of them myself with him, and we are very happy that he's taken the time out to share some of his insights about this. And he will introduce Dr. Viswanath, and he will take over the proceedings of the evening from this point onwards. So, Dr. Sarkil, we welcome you, and I invite you to take over the proceedings. Please, over to you. Thank you.
Thank you very much, Dr. Rashmin, and I welcome you all to today's session of PGCME lecture series. And we have with us none other than Biju Viswanath, who is an additional professor of psychiatry at the National Institute of Mental Health and Neurosciences. He is also a physician scientist with a PhD in the area in which he's going to speak today, that is in the area of cellular models for psychiatry. He has recently been awarded an intermediate fellowship of the DBT Welcome India Alliance and is a coordinating investigator of the accelerated program for discovery in brain disorders using stem cells. So basically, most of his area has been related to what he's going to speak on. So I think it would definitely be a good experience for all the listeners, because this is one area which is very close to the basic sciences, and probably this is one area which is going to unravel various mysteries in relation to the nosological boundaries of psychiatric disorders. His areas of interest include the biological basis of drug response in psychiatry, especially lithium in bipolar disorder. So you see, whatever knowledge I have about cellular models of psychiatry is primarily based on articles, and that too many of which has been co-authored or authored by Biju Viswanath himself. So I think it is always better to hear from the expert rather than me wasting time on it. Over to the speaker, Biju Viswanath, for the deliberation. Over to you, sir.
Thank you, Sujit. I'm audible clearly?
Yeah, you are clearly audible.
Okay, perfect. Okay, I think I will hopefully take around 30-40 minutes, and then we can have some discussion.
Yeah, sure.
Let's see how it goes. Okay, so I've taken the liberty to expand the scope of the topic because cellular models itself are not, the topic itself is not valid unless you know why you want to do this. So jumping into that. Okay. So some basic definitions while we start. So as we know, a disease, from what we have learned in medicine, is a biological event. and which is characterized by anatomical changes, physiological changes, or biochemical changes. Usually a mixture of these. So in psychiatry, we don't deal generally with diseases. We deal with disorders, which is essentially a clinically recognizable set of symptoms or behavior associated with distress and dysfunction. So it's very important to understand this distinction, because we are trying to understand psychiatry. Although right now we understand them as disorders, we are trying to understand the disease processes which are related to these psychiatric disorders. Now, the other key term that we use very much in genetics is phenotype. So a genotype is the genetic material, while a phenotype is a set of observable characteristics of an individual, which could be anything from height to hair color to disease. Right. Now, why do we want to study mental illnesses? Of course, this is a psychiatric lecture series, so I think this is redundant. But then, we need to understand why, and sometimes we need to make people understand why. So 1 in 7 Indians affected by a mental disorder. So this is 200 million individuals, and this amounts to 15% of total years lived with disability. So as we know, depression and anxiety constitutes a majority of its psychosis, and childhood development disorders are still a substantial proportion. Now, ours is a young population, and so as we age, the dementia-related burden is expected to increase. The mortality is not just because of suicide, but because of medical reasons. And of course, there is a lot of stigma, and people don't get medical treatment as they should. So stigma is a major problem in psychiatry. And when I started this work, I was kind of hoping that understanding the biology of psychiatric illnesses would actually help in reducing the stigma. So this is again very basic things about affect behavior and cognition, and we know that most psychiatric illnesses fall into the spectrums of either none of these are developed in childhood development disorders, or part of it is developed. As we grow old, we lose some of these functions and leads to neurodegeneration. Because there is schizophrenia where these parts don't work in harmony or concordance. And this is a typical example of an artist who was fascinated by cats, and he develops schizophrenia. And you can see the nature of the cat essentially determines his perceptual problems. A mood disorder is a qualitative change of mood from sadness to ecstasy, but we do find that the thoughts and actions are considered understandable with respect to the mood. And this is again an example of a composer who had multiple suicide attempts, who had multiple depressive episodes and hypomanic episodes. And as you can see, these are the number of compositions that he made during these episodes, and this clearly mirrors a kind of a life chart of a bipolar disorder patient. Addictions when we cannot stop substances, and OCD when there are repetitive thoughts and actions. Now, what is important to understand that all these illnesses in psychiatry, they're highly comorbid. It is not that only one type of disease happens in one person. There are symptoms that occur which are related to some other disorder as well. Now, the other thing that we need to know is that treatments are mostly symptomatic. So we don't have disease-modifying treatments. And without understanding the biology, we cannot have disease-modifying treatment. And that is one reason why research on to the fundamental mechanisms is very important. Okay, now I'm trying to make the point here from here on that a lot of the psychiatric illnesses are neurodevelopmental, which essentially means the development of the brain is altered in many of these illnesses. So this is a very famous or popular cohort, the Dunedin cohort from New Zealand, and where we find that if you look at the figure on the left, we find that I don't know if I can. Okay, is my cursor visible? I hope it is visible, but anyway. So is my cursor visible, Dr. Sujit?
Yes, it's visible.
Okay. Yeah, sorry. So as we can see that among the 70% of mental illnesses actually have an onset prior to the age of 25 years. And many of them, 3/4 of them actually had a first diagnosis, or many of them had symptoms which were predating it. Essentially 25 to 60% of cases have a history of childhood difficulties. Now, this is a big number that we are talking about. This is coming from a population cohort. Now, essentially indicating that phases of brain development, cell birth, migration, differentiation, maturation, synaptogenesis, synaptic pruning, which occurs in adolescence, and myelination, which happens for a long time into adult life, is altered in these illnesses. Now, giving an example, this is an early study from the group, from Dr. Benegal, where he actually examined people who have not had illness, but these are family members, unaffected children of people with mental illness. And here he finds smaller volumes of multiple brain areas. And you can see these are very important areas: hippocampus, amygdala, frontal gyrus. And you can see that these changes are happening much earlier than the onset of illness in these people, indicating that there are neurodevelopmental changes in the brain which are happening. Now, this is a study which has examined people who have a genetic polymorphism related to Alzheimer's disease. And these are young people, 50 years before the onset of Alzheimer's, young people, and they have brain changes which are related to, which are changes which are not seen in people who do not have this variant, indicating that some of these changes are happening much earlier in life, and that is evident even for neurodegenerative conditions. And most of this is genetically determined. Right. So they're making the point that adult psychiatric disorders rarely arise de novo without any warning. Most patients and families, if you are very careful, will recall difficulties in cognitive, emotional, behavioral problems from childhood. Now, they may lie dormant till a particular point, and then at some point, an environmental stressor might be there to precipitate the onset. This is essentially the neurodevelopmental continuum of psychiatric illness. Okay, so what do we understand about psychiatric illness at this point? What we do understand is for many of the major psychiatric illnesses, there is a clear genetic component, and this is higher for some conditions and lower for some conditions, which I will discuss. But essentially, these genetic components lead to changes in brain gene expression and changes in the brain. There are environmental factors, which I will also discuss, which alter this process. Finally, we have a vulnerable brain in many of these individuals, and at some point, the triggering of the illness happens, and these people have various illnesses that we usually see in the clinic. We'll start with what we know about the genetic variations leading to brain change. Now, I will not go to the details of this. We know the genetic material is important for human beings, and that determines all the biology. Now, these few terms which are very important here is a single. So, as we know, most human beings are similar in terms of genetic makeup. Around 99% is similar, and the 1% makes all the difference. And these are contributed by variations in the genome. Now, single nucleotide variations, for example, point mutations, which are rare, or these could be single nucleotide changes, which are very common, like single nucleotide polymorphisms, which will come up in the next slide. And there are many other kinds of variations, like structural variations, which are considered very important for psychiatry, especially for schizophrenia now, copy number variations, where a chunk of chromosome is repeated. Then there are tandem repeat variations, and these are important for DNA fingerprinting and identification. Okay, so say SNPs. So SNPs are very common. These occur throughout the human. Essentially, it is a change happening in a single nucleotide, and they happen 1 in every 300 nucleotide base pair. So essentially, there are 10 million SNPs within the 3 billion nucleotide human genome. So to be classified as an SNP, it should be present in at least 1% of the population. So there is no. So a mutation is generally considered rare, disease-causing. SNPs are common and not necessarily disease-causing. So if you go to PubMed or Medline, these are denoted by a reference SNP numbers or RS numbers, which is, if you read any paper with genetics, you will find these RS numbers. Now, what is a haplotype? A haplotype is a combination of SNPs happening in a stretch of DNA. So these are 3 SNPs here in 3 colors, T, A, and G. But when you have this stretch varying in these individuals, we find there are 8 haplotypes. So although there are 3 SNPs, there are 8 haplotypes because of the various combinations which are possible through these SNPs. Okay, to keep it very simple here, so in any individual, one part of the chromosome, there are 2 for any homologous pair of chromosomes. One is coming from the male parent, one is coming from the female parent. So it's a paternal homolog and a maternal homolog. What's the genetic locus? It is the location of a particular gene on the chromosome. And at each locus, an individual can have 2 alleles, 1 on each chromosome. So this could be like a capital A, which is homozygous dominant, a small b, which is homozygous recessive, or a capital C, small c, which is heterozygous. Okay. Now, another important term, if you're reading any papers related to genetics, is minor allele frequency. That is the frequency at which the second most common allele occurs in a population. Now, in this case, the second most common allele is C, small c, or small a here. So if small a is found in 10% of individuals, the minor allele frequency is 10 by 100, which is 0.1. If it is 1%, then it is 1 by 100, which is 0.01. That's a very important term to understand. So we all know about case control association study. So a genetic association study is very similar to a case control association study. It is just that the risk that we are looking for here is a genetic variant. So if one type of the variant or one allele is more frequent in people with a disease, then the SNP is said to be associated with the disease. Okay, so I think, okay. Now, this is just showing the different genotyping methods which are available right now. These are very commonly done by all the groups which are doing genetics, both in India and abroad. And so here we can see that on the x-axis, we are determining the allele frequency that we spoke about, right? The minor allele frequency. And here we determine penetrance. What is penetrance? Penetrance just means that if 100 people have this particular variant, will all 100 people have the trait or the phenotype? If all 100 people have, then we say it is 100% penetrance, which essentially means that if the variant is there, the person will have that particular trait. Example is presenilin 1 in Alzheimer's dementia. So all of you would have read about it. If you have a presenilin 1 disease-causing variant, the person will have Alzheimer's dementia before the age of 50. This is 100% penetrance. But most variants which are identified in psychiatry do not fall into this category, and they fall into what we call common variants. So essentially what we are trying to say is that you need multiple variants to come together, which is polygenic causation, to lead to psychiatric illness. Now, there are also methods in which people are now figuring out not just very highly penetrant rare variants, where people are also identifying common variants in large numbers of samples, and there are also people who are finding variants with intermediate penetrance. So there is risk, but it is not 100%. It is less, but it is not very low as also. So most of these methods for finding genes or variants which are highly penetrant are either linkage studies or whole genome sequencing, whole exome sequencing studies. And for the genome-wide association studies, people use what we call SNP chips. I'll give examples of both, which we have done. Okay, so this is one of the families which I'm sure most of you should know. This is the family in which the DISC1 gene was identified. So DISC1 translocation was identified in this family. Now, we typically say that the DISC1 gene is related to schizophrenia, but we do need to understand that this family has people with schizophrenia, with recurrent major depression, with bipolar affective disorder, and other minor diagnoses, which is essentially anxiety disorder, indicating that this is essentially a risk gene for psychiatry and not just for schizophrenia. But again, this was found in a big family. Similarly, we have been able to do this kind of work in South India, and we have been able to do identify genes KUNG4 and CAP2. These are related to actin, which, and as you can see, in a very big pedigree of patients with bipolar disorder with around 30-40 individuals were affected. So this uses whole exome sequencing to identify rare, highly penetrant variants which can cause disease. So in a big family or in a family, what is the risk for a person to have the disease? Let's say if another person is affected. So if, for example, if it's a monozygotic twin, this for schizophrenia, the risk of this person having a monozygotic twin having illness with the other one having schizophrenia is around 48%. And then it goes down. For any kind of first degree relative, it is around 10%. And then it goes down. So heritability, which is essentially the proportion of the variants, which is essentially accounted by genes, this can be measured by using multiple kinds of studies, including the twin, what we call twin studies. As you can see, the heritability for many of these illnesses, that is autism spectrum disorders, schizophrenia, and bipolar disorder, ADHD, very high. These are in the range of 0.8, which indicates that 80% of the variance is actually explained by genetics. So it's very important to understand this genetics. And again, this has been done a lot by genome-wide association studies. So what we do here, so what has been done here? Hello, am I audible? Any issues? Sujit, am I audible?
Yes, you are audible. Yes, Biju.
Yeah, because there's some disturbance, so I'm wondering if something else.
No, it's
okay.
All right.
Okay, right. So genome-wide association studies, as previously mentioned, are looking for polygenic association. So they are looking for common variants, and many gene variants together lead to disease. And to identify this, however, you need huge sample size. And it was earlier identified in around 2010s that one cannot do these kind of studies in small labs and in small parts of the world. You need to have big consortia. So the Psychiatric Genome-Wide Association or Psychiatric Genetics Consortium came into being. And as you can see here on the x-axis is the thousands of cases. So we are talking about 10,000, 20,000, 30,000, 50,000 cases. These are genome-wide significant SNPs. Essentially, these are the significant hits. And as you can see, around 20,000 cases, and then from there on, the number of hits go up. So it's an inflection point. So you need at least 20,000 cases to start studying psychiatric illnesses like schizophrenia and bipolar disorder. Now, the Psychiatric Consortia is huge. We also joined the consortia. Sujit is also part of it. I'm also part of it. We have 900 investigators from 40 countries in this consortia. And these are some of the examples, some of the papers that you should all read. Key studies from the Psychiatric Genetics Consortia, where they have identified the genetics of schizophrenia and they have identified genetic loci related to bipolar disorder. Again, you can see the number of cases on the right side. So what we find here is that the heritability of many of these disorders, schizophrenia, bipolar disorder, ADHD, is very high. Of course, it is lower for some of the other illnesses, slightly lower for OCD, again, lower for major depression. Of course, there are a lot of other environmental factors which contribute to major depression. Now, the other thing that we need to understand is that, as previously mentioned, the comorbidities are high. Similarly, the genetic basis is also shared. So you can find that there is, all of these studies have also identified that there is shared pathology or shared genetics across major psychiatric. So we have also joined this consortia now with a big consortia from our side in India. That's the India Consortium on Mental Illness Genetics in the Clinic with 12 clinical sites. And of course, Sujit is part of it from Kolkata. Now, what these studies have found is that there are major genetic factors related to these pathways which have been identified, which obviously neurodevelopmental genes, because as previously stated, many of these processes are neurodevelopmental. Neurotransmitter genes to a lesser extent, calcium signaling, which is a very important second messenger, synaptic proteins. Now here I will mention the Fragile X mental retardation protein, which has been implicated also not just in autism, but also in schizophrenia. And I will give you some evidence of that. We have genes related to cytoskeleton dynamics, adhesion molecules, immune molecules, and of course, translation and transcription. Right. Now, so we were discussing the genetic factors. Now we'll switch on to another aspect of etiology, which is environmental factors. Now, the exposome. The exposome is a measure of all exposures of an individual in the lifetime. Now, the brain is particularly sensitive to perinatal microenvironment during early development. It's very important to get that history as well. And so, an individual's exposure begins much before birth and includes exposures, psychosocial stressors, diet, lifestyles, and behavior. Now, these are some of the examples: parenting, prenatal microenvironmental, parenting, attachment, school climate, physical environment exposures, adverse childhood experiences, psychological stress, of course, cannabis. I'll give an example of adverse childhood experiences from the studies that we have done. So this, of course, would affect the immune system, the hypothalamo-pituitary axis, and of course, epigenetic modifications, which is essentially changes in gene expression. So what we found here in this study is that we examined, I think, around 600 individuals from families from a very large study that we were doing. And we found that adverse childhood experiences were experienced by at least 2/3 of the sample. That's a big number. And this, again, brought forward the onset of psychiatric illness. This is more, I think, in the men. So essentially, especially more with OCD and substance use, that if you are exposed to adverse childhood events, you are likely to have the illness at an earlier age. What we also found then, we also try to understand whether it changes cognition or neuropsych deficits. And we find here that people who have a familial risk versus people who don't have familial risk, there's a difference. So if you have a familial risk, and if you also are exposed to adverse childhood experiences, you are likely to develop the illness much earlier, while otherwise you are actually resilient. So this was true for actually schizophrenia, bipolar disorder, OCD, and substance. Right. Now, all of these, whether it's genetics, whether it's environment, will affect the brain and finally lead to a vulnerable brain leading to disease at some point. Now, how do we understand these brain mechanisms and brain changes which are happening in illness? But to do this, this study, which I've been part of in the last 5-6 years, funded by the DBT, this is the accelerated program for discovery in brain disorders using stem cell. Here, what we've been doing is that we've been collecting families. So these are families with multiple people who have mental illness. So we have collected, I think, around 500 families right now. And what we do with them, other than the detailed clinical assessments, MRIs, and all that, we also collect biomaterial. We generate cell lines, and we have a DNA repository. So I'll come to the details of that in a moment. So this is a cell culture lab that we have at NIMHANS, where some of this work is going on. And here, what we do is that we are looking at cellular models, which is essentially a disease in dish model that we are following. Now, what we are doing here is that to identify fundamental mechanisms related to the identified genetic variants, we need to have model systems. Now, animal models obviously have disadvantages because these are animals. Now, the use of human cell cultures as an appropriate surrogate is what we do. What we do here is that we take blood, from the blood, we convert them into what we call lymphoblastoid cell lines. This essentially then gets converted to pluripotent stem cells, and then from the stem cells, we can generate neurons, astrocytes, oligodendrocytes, mini brains, and study them. So this essentially shows the pipeline here. So we have done this for 2000. We have around 2000 samples in the bank. And what we have done here is that we take lymphocytes and convert them using Epstein-Barr virus to lymphoblastoid cell lines. So these are immortalized. From there on, these are electroporated with plasmids, and these are called Yamanaka factors because this is the Nobel Prize-winning scientist who found the method of converting any somatic cell into a stem cell. Was Shinya Yamanaka, and this was 2009. And so these are electroporated with these plasmids containing Yamanaka factors, and over a period of a month, these get converted to induced pluripotent stem cells. Can you hear me? I have lost internet. Have I lost internet?
Yes, you are audible. Yes.
I'm still audible? Okay, fine. There was a power outage. I'm just trying to see. Okay, I'm still audible, right?
Yes.
Okay. Hopefully it should be fine. Okay, so this is giving an example of an induced pluripotent stem cell. So you can see this is the stem cell colony where you have cells from an individual. So from the blood, it has been converted to a stem cell. And as you know, a stem cell can become any kind of cell. So this is a colony of cells. And these are immunolabeled. Essentially, we are looking for an antigen related to pluripotency. And you can see that most of these cells are lighting up under the microscope. You can see my cursor, right? You can also see my cursor.
Yes.
Okay. Right. So with these stem cells, what one is able to do, is that you are able to convert. So, if you go back to your embryology textbooks, you will understand that we all started as totipotent cells, the embryo, the zygote, and then onwards. So during neural development, then with various pathways, these cells get converted to various brain parts. So they become the forebrain using some of these pathways, BMP and the Wnt pathways. And then you have caudalization, we have the sonic hedgehog pathways. There are multiple pathways. The current embryology textbooks are quite detailed with these details. So these can be converted. So these pluripotent stem cells with the same techniques can be converted to any kind of cell. They can be converted to forebrain cells, cortical excitatory neurons. They can. I think my cursor is okay. Is my cursor visible?
I can't see your. Yeah. Yes. Right.
Okay. So these can be converted to forebrain cells, cortical excitatory neurons, cortical interneurons, which are GABAergic. These can be converted to retinal cells. These can be converted to midbrain and dopaminergic neurons, cerebellar neurons, and of course, we can convert them into astrocytes and oligodendrocytes. So it is well known now that it is not just the neurons that matter. The astrocytes matter because they are very important for metabolic support. Oligodendrocytes matter. They are important for metabolic support as well. And of course, they're important for myelination. There are important roles of glia in the brain, which have been identified in the last few years. So there's a lot of research going on about identifying the roles of glia in psychiatric illness as well. So this, is my screen completely visible, Sujit? Or is there an issue?
Yes.
Okay. Right. So this is essentially the protocol for converting pluripotent stem cells to neurons. Now, this is like advanced cookery in that sense. You're going to use various factors which are various factors which will activate various pathways which are otherwise active in an embryonic brain, to convert stem cells into brain cells. So here you can see the stem cells. You can see that these are the neuronal bodies, that we call the embryo bodies, neural rosettes. From there on, you convert them into neurons, which stain very beautifully for various neuronal markers. TUJ1, MAP2, V-GLUT is glutamate. So essentially, we are saying that these are glutamatergic neurons, cortical excitatory glutamatergic neurons that we are seeing here in this microscopy image. Now, the other thing we can do is to do calcium imaging in these cells. So you can see these videos. Is the video playing, Sujit?
Yes.
Sorry. Yes. Okay, so we are seeing calcium images. So this is done with Dr. Raghu in the National Center for Biological Sciences, where the calcium currents in these neurons is being examined. And you can see as the neurons become older, 7 days, 14 days, 21 days, 45 days, the number of neuron spikes keeps going up. Now this is from Dr. Shona Chatterjee lab, where you're looking for action potentials in these neurons. So you can see a needle here, which is inserted into the cell body of a neuron, and you can actually measure the action potentials, like what you do in an animal brain. In physiology, you can do it with a human cell. Right. Now the other thing, we have protocols for converting these cells into astrocytes. These are much longer, 6 months. So you can see a similar protocol in that sense. The pathways are different, and you can get these beautiful astrocytes, which you can measure and examine in the dish. Now these are co-cultures of neurons and astrocytes. The green colored cells are the neurons, the orange colored cells, the astrocytes. So you can see that these are cells which are interacting with each other and growing together. Now this is a protocol for conversion of oligodendrocytes. Just give me a second. Okay. Right. So these are technically oligodendrocytes which are expressing myelin. So the yellow colored or the green colored color marker is for myelin basic protein. And so you can see that this is essentially a bed of neurons in which the oligodendrocyte is actually now myelinating. So this again shows this in much detail. The neurons are in red, the oligodendrocyte is in yellow, and you can see that this is the myelinating portion of the neuron. You can see that the oligos are trying to myelinate the neurons here. And these are all cultures that we have done in the lab. Of course, these are long-term cultures, and it takes a lot of time and effort from students to get there, but it's very exciting to see. So these are mini brains. So we can also culture them not just as 2D cultures in the dish, but you can also culture them as 3D. So these will be spherical structures, as you can see, and these are the red colored cells are the neurons, and the yellow colored cells are the glia within these spherical mini brains. And these now can be kept for 200 days, 300 days. Right. To give some examples of some of the work that we have done, we first wanted to look at neurodevelopmental genes. So we started looking at FMR1, which is essentially Fragile X mental retardation protein, which further leads to autism. This is part of my PhD work. Let's skip these details. What we find here is that if you generate oligodendrocytes, myelinating oligodendrocytes, from FMR1 null, which essentially is genetic variation which leads to Fragile X mental retardation, and we find that these cells are smaller than the ones which are controlled. So you can see, you can ignore the figure on the right, but essentially you can see that the number of arborizations or the number of branching points in a control is much higher than that of the patient line. And this has been quantified on the right. You can ignore this. Now, what we did, so this is for Fragile X, but then we wanted to do it for psychiatry in bipolar disorder and schizophrenia. So we took a pedigree where we have multiple people actually with bipolar disorder. And here we can see that some of the genes that were implicated by whole exome sequencing were related to neurodevelopment, and these are all related to neuronal migration. So you can see that all these genes, NIPBL, SCN3A, which is a sodium transporter, NRG2, neuregulin, and anilin, all of these are related to cell migration. So we wanted to look at, okay, are these cells migrating differently? Are these neuronal cells migrating differently from the control? And here, so you can see these videos. I hope the videos are visible. You can see on the left is a control cell line, on the right, a patient cell line. The control cell lines are together migrating towards each other, while the patient cell lines are trying to grow, are moving alone. They're not going towards each other. They are technically going all around the place. You can see that on the left, they're growing together, they're coming together, while on the right side, they're not going together. They continue to move, but they're not doing it cohesively. This has been quantified, as you can see from the traces. Now the control lines are going towards each other, while the patient lines are going all around the place. So although the velocity of migration is much higher for a patient line, the effective distance traveled by the patient line is much lower. So this again is something that we are further working on to try and understand what genetic changes are leading to this. Another example where we have not just looked at disease, but we're also looking at drug response. So here we are looking at lithium treatment response. So we had patients who responded to lithium, we have patients who did not respond to lithium. So what we find here is that in the bipolar patients, the mitochondrial activity is lower. We find that the cell death is higher in patients and the cell lines from patients. But what we also interestingly found that if you add lithium, then the cell death in these lines goes down. But this happens only in responders and does not happen in non-responders. Now, this is very exciting to try and understand what is leading to this particular change only in responders and not in non-responders when you add lithium. So again, some of the exciting activities that are hopefully going to happen in the future. So with that, I will stop, and I think I've taken around 40 minutes to give a glimpse of many of these things which are happening. The funding agencies have been very generous with us. Our collaborating institutions have been very generous, the National Center for Biological Sciences and INSTEM, both in Bangalore. We have had funding from the DBT, ICMR, Pratiksha Trust, the India Alliance, which funds me, and of course, a lot of other funding agencies, including NIH and UKRI. I thank the team and I thank the organizers for allowing me to talk. Thank you. Yep.
I think, thank you, Biju. Okay. So, I think there are areas where I think the PGs and the young psychiatrists would be, curious to seek clarifications. And yes, I'm expecting questions now. See, one important thing is that these days it has become almost inevitable to come across articles where most of these areas are dealt with, and often if you are not aware of the basics, often it becomes just mugging up this part. Even in the standard textbooks, the genetic parts, they are coming up, and it is undeniable. And I think that way, the way the lecture proceeded, I think it has enhanced significant clarity. At least my understanding has also been enhanced in certain areas. So I think PGs should be having questions and clarifications. So I don't see, okay, only comments in the chat box. If anyone has any question.
Can people ask, or they can only put it in chat box?
No. If they want,
they can ask,
want, yes. They can unmute and ask, because all are in the same link.
Right.
So we have one in the chat box from Sagar Snehalata Devidas. What is the role that a psychiatry resident, what is the role that a psychiatry resident can play in studying and applying genetics?
Right. Yeah. So at this point, doing psychiatric genetics is quite easy, especially if you're an MD student or a DPM student. You have your thesis time. So a lot of data is available for you through so many channels. The Psychiatric Genetics Consortia leaves most of the data open for people to use. It's very easy to get access to them, especially people are very happy if someone from India, for example, wants to participate, because there are many opportunities. We have collaborators who would be happy to train anyone who wants to work in the field. And data is available. So I don't think that will be a problem. So that will be one way to start. If someone wants to, work in the field, because trying to generate data when you are an MD student is a lot of work. But if you learn to use the existing data, that itself is a very powerful thing to do. I think that will be the start. If I was an MD student now, that is what I would do. I will use available data, which was not available, let's say, 10 years ago, when I was, finishing up my MD. I didn't have it. So that's a luxury. So that's what I would. I don't know if I answered your question. Please feel free to talk. Don't just chat.
Yeah, I think that was, I think that has answered the question. So we have a question from Dr. Prasad Rao, sir. Sir has asked, can we have evidence to use the tests you got evidence as biomarkers? So can we actually, he means that whether we can use these tests, have we got enough evidence to use any of these tests that you discussed as biomarkers?
Yeah. So for anything to be a biomarker, you will need it to be done in thousands of individuals, and and then again, it needs to be something which you can do easily. Let's say in blood. So most of these experiments are done with after generating stem cells and then generating neurons. So that's a long way, down the line. And so to get any leads from there and go back to blood, of course, it will take a lot of time. So it's still very much in the infancy stage of work in psychiatry. I think only the last few years has this become, a thing in psychiatry in that sense. So the project that I discussed about the ADBS, I think came up in one of the, so that is a major project which is trying to work on it, because that generates a biorepository. Around 100 cell lines have been generated. They do training workshops to train people who can start using those cell lines. Plus there's a repository of DNA, and a lot of work going towards having. So I think, for example, the Gentis consortia that we are both part of, we are trying to, I think, sample 20,000 individuals as part of that project. And so we are kind of hoping that with large-scale studies, both deeper and broader understanding will be there, and that will hopefully lead to biomarkers, both for not for disease, but also for drug response and stuff like that. It will take some time.
I think sir also has another question. Is SNP studies in psychiatric genetic studies obsolete? So is SNP studies?
That depends on, if you're looking at single SNP studies, yeah, they seem to be obsolete right now. Because single SNP studies in itself are not well powered to identify a change. So it leads to false positives. But then what people do is genome-wide association studies. So essentially you're looking at the whole genome, and then you're looking for changes happening across. So which is why the most recent study, I think, with schizophrenia, which I have not actually shown today, I think has used around samples from 67,000 people. And they've identified hits from SNPs, 271 SNPs, which are important for schizophrenia. So single SNP studies, probably obsolete. But large-scale SNP studies is the way to go at this point. GWAS and other kinds of studies.
Okay, thanks, Biju. There is another question in the chat box from Nechili Thodi. I don't know whether I pronounced it correctly. Do larger brain people have bigger IQ? Is there a method to increase brain reserves by increasing cell size? Do people with larger brains have larger brain reserve?
I don't know. I'm not the expert in saying this, but I don't think so. I think the simple answer is no. I don't think it has anything to do with size. Probably it has to do with connectivity, but then again, that again needs to be answered by someone who has expertise in MRI imaging. But yeah, simple answer is no, I think.
Fine. Anyone else who has a question? Or maybe comments?
If I may be permitted
yeah,
to ask
sure, please.
Please.
Dr.
Biju, you're known to have an interest in the biological base of drugs, you had mentioned to me, and particularly lithium. So I'd like to know a little bit more about it, if you can share your insights and experience.
I mean, so that's an area of active research. There are, I think that kind of work has been happening for the last 6 or 7 years, when people have studied people who are responding to lithium and people who are not responding to lithium. What people have identified is that there seems to be a physiological readout in the sense that lithium seems to attenuate certain physiological, hyperactive neurons, which happens only in the responder group. Now, why that is happening is again being researched very actively. There are at least 2-3 major groups in the world who are this group by Fred Gage in the US, and that's the group by Ivan Schneider in, I think, Europe, someplace in Europe, I think Germany, who are examining this. We are also trying to do this in a small way. We are trying to understand whether these changes are happening because problem with lithium has too many targets, and it is water soluble, and it will go everywhere. It's non-metabolized, and it will hit so many pathways. Of course, the more famous, popular ones, the inositol targets and the other second messenger systems, calcium signaling, and all that, GSK3 beta. But then we don't know whether any of these is actually leading to the clinical changes, and so that is something which is actively investigated. But again, only for the last 6 or 7 years. Very much in progress. I don't know if I answered you. Please feel free to clarify.
Can you give us a simpler example of a drug which probably is not as widespread or which is easier to use as a sample for explaining how the biological mechanisms might be explained on the basis of the cellular models?
Okay, so people have studied the effect of serotonin as a neurotransmitter, right? I mean, that's as simple as it can get. But then it's not as simple because serotonin is very important for very many things, starting from early development. So people have specifically studied effects of serotonin on the mitochondria. So one of my collaborators in the Tata Institute of Fundamental Research, Mumbai, Dr. Vidita Vaidya, so she has actively studied the effects of how serotonin increases the generation of mitochondria in the cell. Okay, and that is a very valuable form of research. So now we are trying to understand whether, for example, SSRIs, which we use in depression, OCD, work in the same manner. Is that simpler?
Yes, sense? Of course. Yes.
Yeah. But yeah, it's exciting. But then the problem is it's never simple in psychiatry, because psychiatric drugs, when I say lithium is complex, lithium is not as complex as many of the other drugs, because all your antipsychotics, all your antidepressants work on so many pathways, right? And so many things are altered, and there are so many targets. And because there is an additional issue that they are metabolized, and that issue is not there with lithium. So actually studying lithium is simpler. Studying some of the others are more complicated. But yeah, it's exciting.
But then, if I may, where do you see this going forward? How does this change our way of dealing with psychiatric illnesses and the treatments for treatment options for psychiatric illnesses on the back of whatever your research is indicating to us and whatever it might probably point
in
to
multiple, yeah, so in multiple ways. For example, we are also trying to understand why clozapine works, right? Now, clozapine is such an important drug, and it works for people who have not responded to anything else. Now, if you know how clozapine works and how specifically clozapine works, that may make a huge difference. Now, one way to go about it is take a person's cell line who responded to clozapine, put clozapine, but then also put all the drugs which are not worked. Okay, you're trying to understand what is specifically clozapine doing, what the others are not doing. Now that will definitely, if you know how clozapine works at this point, that will make a difference, right? You can try to make a new drug. Of course, these are all, one can say these are all very time-consuming process, but now with the advent of what happened with COVID-19, we know anything is possible, and that too in a shorter time. So, any new discovery in the field has not happened for many, many, many years now, for many decades now. So, any new discovery, I think, will have far-reaching consequences, either in terms of identifying new drugs, new targets, and when drugs with lesser side effects. I think these are all important.
Okay. Excellent. Yeah. Thank you.
Yeah. Just taking a cue isn't, yeah, I think someone wanted to speak, please. Someone unmuted? Okay, I actually just wanted to just raise a topic, Biju, whether you see all this work that you people are doing and in collusion with other people in the field of basic science research in psychiatry. So do you see it, I mean, just maybe at the level of speculation or too early to ask, but do you actually see it, as we often keep reading, do you actually see it changing the classificatory system or the nosology drastically in maybe another 20, 30 years or so?
So it is already changing. Say, for example, if you look at any genetic study right now, bipolar disorder and schizophrenia are considered in the same breath. So everyone knows, anyone in the field of genetics knows very clearly that there's high genetic correlation between schizophrenia and psychotic bipolar. Okay. So, sorry, I can't hear you. Hello? Shraddha?
No. She has accidentally, I think, unmuted herself.
Okay.
Yeah, please go
Right.
on.
So what I'm saying is that, of course, as we find new things, so the moment you find anything related to the biology of any of these illnesses, I'm sure a new diagnostic system will come up with related to that illness, right? I mean, that will keep changing. So there are illnesses which are, the moment you know that it is related to a particular gene, it is related to a particular system, a particular pathway, it will definitely affect diagnostic systems for sure. Hopefully for the good.
Yeah.
Like even now we say, right, schizophrenia is a group of schizophrenia. I mean, it's not just one kind of illness. There are so many types of illness which come together. Same thing for OCD. So, I mean, so many varieties, and I mean, the heterogeneity is so huge for many of these illnesses. So yes, I definitely envisage a change which will happen, hopefully earlier than later.
I think that has been a quite fruitful discussion to begin with, and I hope this lecture and subsequent discussions will generate interest among the listeners, and especially the PGs who might be interested in taking up research in this area, because as Biju was saying, that we need to develop a consortium approach to have more and more patients to further genetic research in this area of psychiatric genetics. And I think lectures like this would stimulate this process or mark the beginning of interest in these areas. And I would definitely thank Dr. Rashmi and Dr. Biju for having chosen this topic. And I think those who have missed it, it would probably be available in the form of a recording. So before I conclude the session and hand over to Dr. Rashmi, is there anyone who would have any question or clarification? Over to you, Dr. Rashmi.
Yep. Thank you. That was an excellent talk, and it again and again reiterates the fact that the more we learn, the less we know, and we have been exploring newer and newer areas in the field of psychiatry and the cellular model of psychiatry and the biological mechanisms that we understand with the help of these models is really very exciting. And we are very grateful to a co-researcher like Dr. Viswanath for having taken the time to explain it to us, and it's fantastic. So we're very grateful to him for this, and we hope this would inspire some of the younger brains in the postgraduate group to take this up as a research opportunity, use this as a stimulant for their academic appetite. We hope to have vetted it a little bit, and perhaps we'll try and do that more often in the future. We need young researchers to go and tread the path which people like Dr. Viswanath are walking on, and that's the way psychiatry and the understanding of neuropsychiatry is going to grow in the future. So thank you very much, Dr. Viswanath, and thank you so much, Sujit. You were very kind enough to have done this, and you steered it along quite nicely. And of course, the audience who took their time out and are with us today to share this vast area of knowledge, but in a tidbit manner in a 45-minute session. So it was a teaser, and we are happy that you were with us. And of course, I'd be remiss to not thank Sun Pharma for having supported us throughout this entire academic journey of us. So thank you all once again. Thank you, Dr. Biju Viswanath. Thank you, Dr. Sujit. And thank you all of you for being a part of this. And we hope to see you again next month with another exciting talk from another really good faculty. So thank you all once again, and I wish you a good evening ahead. Thank you.
Thank you.
Bye.
Thank you very
Thank you.
much.
Thank you.